So moving on to the neurobiology of addiction, as I mentioned, I'll briefly discuss 4 neurotransmitters that have been related to addiction, and also we have evidence suggesting that exercise can affect these neurotransmitters. I do want to point out that each one of these neurotransmitters and their role in substance abuse and exercise could each be their own individual presentations. So I apologize in advance that I may not be able to provide a complete picture of the role of each of these, but hopefully I can provide enough evidence that would suggest that there are effects of these neurotransmitters in the development and relapse and reinstatement of substance abuse, and that exercise also can affect these neurotransmitters. So I will begin speaking with about dopamine. Dopamine is the neurotransmitter that's most commonly associated with substance use. It's the mesocorticolimbic dopamine system is typically referred to as the brain's reward center. Stimulants such as cocaine and methamphetamine activate the circuitry of the dopamine system. And we see that in animals, if the dopamine system is destroyed either through a neurotoxin or surgical lesions, it results in a reduction in the reinforcing effects of stimulants. Acutely, cocaine works on the dopaminergic system by blocking dopamine transporters. You'll see here this is a dopamine transporter that has been blocked by cocaine. It results in increased dopamine in the synaptic cleft. This acute occupancy of the dopamine transporter is associated with subjective high reported by individuals who have been administered cocaine. And we also see that density of the D2 dopamine receptors are inversely associated with the reinforcing effects of cocaine. When we look at the role of dopamine in drug craving, we see that drug-related cues increase dopamine in the dorsal striatum, and this is associated with an increase in cocaine craving. And you can see correlations here. This is occupancy of the D2 dopamine receptor and its relationship with self-reported craving. When we look at the role of dopamine in the reinstatement of drug-seeking behaviors, we see that cocaine-triggered reinstatement of cocaine results in an increase in dopamine that's associated with a reinstatement of that behavior. So that's a drug-primed cue. And then we also see that D3 receptor antagonists inhibit drug-seeking behaviors following both drug cues and stress cues. So we see that dopamine seems to play a role in all 3 triggers of relapse, the priming, the environmental cues, and stress. And then very quickly, this is probably an area that most people are more familiar with. So as you see as I go through this, I'll spend a little bit more time on the role in cocaine, and then briefly talk about dopamine and exercise. The effects of exercise on these systems are hopefully at least a bit more familiar to everyone here than the effects of the stimulants. So in mice, we see an increased dopamine turnover following acute bouts of running. In humans, we see an increase in dopamine in plasma. However, PET studies that have been conducted following an acute bout of exercise find no significant changes in synaptic dopamine. When we look at chronic effects, I'm not currently aware of studies in humans that have looked at chronic effects on the dopaminergic system. However, we do have a few studies in animals, and we see increased density of the D2 dopamine receptor. And in animals that are aging, we typically see an age-related decrease in the D2 dopamine receptor density, and that chronic wheel running can prevent or at least attenuate that reduction in dopamine receptor. We'll move on next to serotonin. Again, we'll start with the acute effects of stimulants on the serotonergic system. Acute administration of cocaine results in an increased reuptake of serotonin. We also see in animals that fluoxetine, which is an SSRI, can reduce the rewarding and reinforcing effects of cocaine administration. And the depletion of extracellular serotonin increases the rewarding effect of cocaine. And so here's a graph that shows the increase. This shows serotonin and also dopamine. So you see the increase in both with self-administration of cocaine. When the cocaine is removed, you see the reduction, and associated with withdrawal is a drop in both dopamine and serotonin below the baseline levels. Over time, we see that repeated cocaine exposure can decrease synaptic availability of serotonin. When we look at the role of serotonin in drug-seeking behavior, we see that exposure to drug-related cues can result in a decrease in serotonin in the prefrontal cortex. Again, following drug-related cues, administration of a 5-HT1BA receptor agonist can attenuate cocaine-seeking behaviors, and a 5-HT3 antagonist administered during withdrawal has been shown to inhibit reinstatement of cocaine self-administration. So as opposed to what we saw with dopamine, I'm unaware of any studies that have looked at the role of serotonin in either stress-induced or priming-induced reinstatement of drug-seeking behaviors. With the availability of SSRIs, there have been a few studies that have looked at the efficacy of SSRIs in the treatment of cocaine abuse. In rats, we see that pretreatment of fluoxetine can reduce self-administration. However, in humans, the results are inconclusive. One potential explanation for this is the difference in SSRIs that were used. Citalopram is more selective, targeting only the serotonin transporter gene, where fluoxetine also has affinity for the 5-HT2C receptor, and activation of that receptor has actually been associated with an increase in substance abuse. Quickly moving on to exercise and the effects of exercise on serotonin. In humans, we see a decrease in serum 5-HT following an exercise intervention. This was after a 5-week intervention, I believe. This is actually similar, though counterintuitive perhaps, we see similar reductions in serum serotonin following SSRIs as well. In animals, binding of the serotonin transporter and the 5-HT2A receptors are increased following exercise training, and we also see increases in mRNA of the serotonin transporter as well as the 5-HT1B receptor following chronic wheel running. We'll move on next to the HPA axis and stress response. Acute administration of cocaine results in increased plasma ACTH in rats and increased cortisol in humans. And chronic exposure to cocaine results in decreases in the CRH receptor binding. During self-administration, we see that the HPA axis appears to play a role in learning and self-administration. Increases in plasma corticosterone, increased sensitivity to reinforcing effects, and we see that adrenalectomized rats do not learn to self-administer cocaine. So this would indicate that this plays a very significant role in the acute reinforcing effects of cocaine. Similarly, in animals that have been adrenalectomized, we do not see a reinstatement of drug-seeking behavior following a foot shock. So this is considered to be an environmental stress. However, removal of the adrenal gland appears to have no effect on drug priming reinstatement. So those animals who were primed with cocaine did reinstate drug-seeking behaviors. This further illustrates the role of HPA in reinstatement following foot shock. This is a substance that reduces the synthesis of adrenocorticosteroids, and the CP here is a agonist of the CRH receptor binding. So they both have an effect on drug reinstatement following a foot shock. So again, interesting that that the HPA axis appears to have a role in some types of relapse triggers, but it's not an across-the-board effect. We do have some human research implicating the role of the HPA axis in stimulant use. In humans, stress-induced cocaine craving, so individuals who left treatment and demonstrated, as they left treatment, demonstrated cocaine cravings associated with stress. That was associated with shorter time to relapse. That cocaine craving was associated with stronger cortisol responses to stress. And so here are a couple graphs. These are cortisol and corticotrophin responses to stress associated with amount of drug use. And so you'll see that the greater the response of both cortisol and corticotrophin to stress, the greater response is associated with greater substance abuse following treatment. And similarly, when they were dichotomized as either high responders or low responders to stress, you see a greater increase over time, a quicker relapse in those high responding individuals. The HPA axis and the effect of exercise on the HPA axis, this is probably an area where there is a bit of conflicting information, though there is at least some evidence to suggest that exercise can affect HPA axis function in at least certain segments of the population. Exercise does increase cortisol excretion. Chronic exercise training in humans is associated with a reduced cortisol level. And physical fitness has been found to be associated with reduced cortisol response to psychological stress. The final neurotransmitter I'll touch on briefly here is the endocannabinoid system. The endocannabinoid system, one of the ligands present, which I'll discuss in a minute, is anandamide. It binds to the CB1 receptors. So this is an interesting mechanism to look at because the endocannabinoid system appears to not have an effect on the acute reinforcing effects of cocaine. Mice without the CB1 receptors can still learn to self-administer, and blockade of the CB1 receptor does not inhibit cocaine self-administration. But there does appear to be a role of the endocannabinoid system on stimulant use. CB1 receptor antagonists inhibit cocaine and cue-related relapse. However, it does not attenuate stress-invoked relapse. And cannabinoid agonists can provoke relapse to cocaine-seeking behaviors following a withdrawal period. And again, the literature on exercise and endocannabinoids is fairly limited to this point. However, we do see increases in anandamide following acute bouts of exercise, and endurance training can reduce the expression and density of the CB1 receptors. So finally, I'll conclude with a few of the more recent studies that have actually looked in animals at the effects of exercise on cocaine self-administration. So this first study is a study that came out just a couple years ago. You have sedentary mice and exercising mice, and what we see is the reinforcing effects are not eliminated, but they are attenuated following exercise. So we have 2 doses of exercise plus saline solution, and the amount of responding or lever presses. And you can see that they increase with an increased dose of cocaine, but that in both groups, but that at both doses, the exercising rats responded less, suggesting that the reinforcing effects were at least attenuated. Exercise has also been used and demonstrated to reduce substance priming-induced reinstatement. So we have 4 groups here. The first, or the bottom here, is an animal that their running wheel was locked the entire period of the study. Here, the animal only had access to the wheel during extinction. The dark bar, the animals had access to the running wheel during both extinction and during reinstatement. And then the top bar here were animals that only had access to the running wheel during the reinstatement period. And so here we see with greater doses of cocaine, we find some significant results. What we find is that exercise was necessary during the reinstatement periods. Those animals that exercised during extinction but then did not have access to wheel running during reinstatement increased their responses in self-administration of cocaine, but those animals that had access to wheel running during the reinstatement period did not reinstate as greatly their drug-seeking behaviors. And the final study, so the last one was looking at drug-induced relapse. This is going to focus on behavioral or environmental cues. And we see these animals were taught to self-administer cocaine over 10 days. Following that 10-day administration period, the cocaine was removed and extinguished over 14 days. The behavior was extinguished in both groups, sedentary and exercise. During that forced abstinence, they had access to wheel running 2 hours a day. And we see that during reinstatement, a significant reduction in reinstatement of drug-seeking behaviors in those exercise animals. So this is slightly different than the previous study I presented in 2 ways. This is looking at an environmental drug-related cue, and these animals did not have access during that reinstatement period to the wheel running. So it appears that the continuation of exercise may be important in reducing the priming-related reinstatement of drug-seeking behaviors, but is not as necessary in reducing the drug-seeking behaviors of a drug-related cue.